Well, thank you. Well, I mean, I will, of course, share some similarity and probably quite a lot of similarity, being how far it is from the sorts of things that have been spoken about already today. And I just want to thank the previous talkers for such far-ranging and interesting ideas. And I am just going to add on a very sort of narrow approach here in comparison. I also had to get all my excuses out now. It's somewhat jet-lagged and hastily put together, so I apologize. One, if it's incomprehensible, or two, if it is comprehensible, but yet has such a working memory load that it's effectively incomprehensible. But anyway, here we go. So what I'm going to be talking about is, why do neurons in the brain look the way they do? Why do they structure themselves the way that they do? And so the sorts of problems that we're going to be thinking about is how neurons structure themselves to allow for flexibility. So what might this mean? It might mean that, just 1 second, let me just start a timer. It might mean that, how do I go cooking? So for example, when I go cooking, I need to go to a fridge, I need to chop up the vegetables and put them in the oven, and then finally I can sit down for dinner. And so that is a single task in its own, but of course it can be broken down into different components. And there's a sort of the spatial element of it, that the kitchen exists in space and things exist in space. So I can sort of understand this overall this task with one component, and that's the spatial component. Of course, there's also the stimuli component of what's going on. There's a fridge, there's an oven, chopping board, and dining table. And then also it's the notion of the task. I need to know that I need to do things sequentially. Of course, if I put things in the oven immediately after I took them out of the fridge, it's a bit of a pickle because I haven't chopped them up yet. So there's 3 components here to this task. And knowing all of them is really quite useful. Decomposing it in this way is really quite useful, because just how kids can make, out of some blocks, they can make new towers, and out of those blocks, we can come across and make a new task out of those blocks. I can learn how to cook in my kitchen, but I can still cook in your kitchen where everything's in a different configuration and the fridge might look different, and so on. And so if I'm able to decompose my task into different subcomponents, I can then recompose them to understand something new. And of course, in probability parlance, this will be particularly applicable when my task itself is factorized, i.e. space, task, and stimuli can occur in any different combination. And then in which case, it's particularly useful to understand that that's a factorization that exists. And of course, the observations I see in the real world will be some function of those of space, tasks, and stimuli. Okay, so there's the thoughts of things we're going to try to understand when we have sort of tasks that we have various factors that we want to generalize. How do we go about learning them? And so it's all very well and good, at least like saying that it's helpful to represent things in terms of building blocks, but how on earth do neurons in the brain or neurons in the machine actually represent these building blocks, or represent the combination of those building blocks? So here, for example, is in space how neurons do represent these building blocks. So what we're showing here is a top-down view of a box, and the black trajectory, sorry, where a rodent has run around in this box. So it's run around in this box, and it's that. And every time you see a red dot, that is where a single cell, we're just looking at the activity of a single cell, where a single cell has fired. Okay, so we're sort of looking at the spatial, where this cell fires in space. And we can turn that into a rate map. And this thing is called a grid cell because it fires in this beautiful hexagonal grid somehow. And it's really a building block of space. So lots of these cells all look very same, and they provide a basis for space. There are also other sorts of cells in this area of the brain called entorhinal cortex, which are the building blocks for objects. They care about this. They fire at certain distances and directions away from objects. So here's one cell on the left, and here's another one that fires to the west of this white object. It's really the building blocks of objects. There's lots of those cells as well. And finally, you also have these sort of building blocks of borders. These are cells which fire at borders or at certain vectorial distances from borders. And so you have these cells which kind of form in these beautiful little submodules of ones that care about space, ones that care about objects, ones that care about borders. And of course, that makes sense of why you'd want this disentangled representation, because objects, borders, and space all occur in different combinations in different rooms. But unfortunately, life isn't quite as clean as that. And if you look inside the brain, sometimes these building blocks sort of blend together. And so, for example, this is originally on the left, this is a grid cell. And after I train it train this animal in an environment for a while where these black dots are now these objects that it likes to run around towards. I should say that I'm not training this. This is someone else's real data. You can see that this grid cell had firing fields in these locations, and they sort of warp towards these objects. And here's one cell, and here's another cell where the firing field has that used to be here is warped towards this other object. This is sort of curious because now we no longer have this perfect dichotomy of grid cells caring about space and these cells caring about objects, but now we've got a cell which is a blend of both of them. It's sort of a building block for space and objects. So that's really rather curious. And of course, there's lots of non-spatial representations in the brain as well that show this similar phenomena. You have cells in parietal cortex, which care about just one task. Here's a cell here that cares only about the T-maze task, doesn't care about this other task at all, where here's a cell down here which only cares about the other task, but doesn't care about the first task. You have this cells that care about specific things, one cell for one thing. And you get that in, and so here's across all of the cells they recorded, most of the cells just lie on this sort of L shape. They just care about one of the two things. And likewise, you also find lots of these sorts of cells in prefrontal cortex. But again, biology is not so clean. You also find these mixed selective cells for non-spatial problems. For example, these cells care about these sort of abstract decision-making tasks, but each cell can care about lots of the different tasks. Okay, so we have this kind of weird situation where sometimes cells care about one thing, just one thing, and sometimes an individual cell care about many things. Okay, that's called disentangled or entangled, or selective or mixed selective. Of course, this isn't just a thing in neuroscience. Machine learners care a lot about this as well. So, for example, in the world is often believed to be the data we see in the world is often believed to be generated from some latent factors. And of course, what machine learners like to do is they like to observe, see this data, and they want to come along and infer what these latent factors might be. And of course, if you can infer what those latent factors are, brilliant. And if it's the case that a single neuron cares about a single factor, even better, because then you can really understand what's going on. This is the neuron that cares about people. Brilliant. I have a bit of an interpretable algorithm at that point. And so what people tend to do is they train these things called autoencoders or variational autoencoders, where they put the observed data in, they put it into some latent representation by some linear or nonlinear transformation, and they have these sort of methods to force that latent layer to be disentangled, to be factorized. Okay, and once they do that, they can see lots of interesting things. So here's, for example, on the left, here's this algorithm which is trained on lots of images of squares, circles, and hearts of different colors and different positions. And we can see this neuron, what we're showing here is there's 10 neurons here in this latent layer, and we're scanning across what this neuron cares about. So this neuron at the top cares about the Y location. This neuron cares about the X location. This one cares about the size. This one cares about the shape, and these ones are the colors, and etc. And these ones don't care about anything. So they've got these disentangled representations, and there's also, you can see it in other data sets as well. And so curiously as well, if you take these models which are really good at learning disentangled representations, and you fit them to brain data, so for example, this is neurons in infratemporal cortex, you can see that single neurons in infratemporal cortex are explained by single factors. So it looks again like these neurons in the brain really are just caring about one thing, one sort of latent data generative cause. So this sort of brings about a lot of questions. The first is, why do sometimes single neurons care about single variables? And why do they sometimes not? Okay, so is there some sort of general principle that isn't just for space and isn't just for brains that can tell us about this? Oh, sorry, I should have been clicking. Okay, so and lastly, why on earth does each cell actually look like it does? So why does a grid cell look like a grid cell? Why do cells forming these modules that care about specific things? These are the sorts of questions that we're going to try to answer. So the first one we're going to try about the first questions. Why do you get these disentangled representations? And then we're going to try to wonder about that a little bit. But all of these questions, what we're going to the way in which we're going to try and wonder about it, so we're really going to think of what are the actual constraints on neural representations, and then we're going to run down that rabbit hole and see if it leads to somewhere good or not. Okay, so what is a constraint on a neuron? Well, first, of course, the neuron actually has to be useful, it has to be functional to something. It needs to represent something in the world of use. So, for example, if we've got some neural representation here, where red's meant to, dark is meant to indicate the neuron is active, it might be useful for it to decode position, where it is in the world. It might also be useful to decode what sort of behavior do I want to do, go east, etc. And of course, a different representation needs to encode a different thing, a different position, a different thing that I might want to do in the world. Okay, so constraint, neurons have to be useful. The other one is they need to obey biology. So for example, neurons in the brain, they do not spike negative. You can't be negative. Okay, so that means that something like this isn't okay, but something like this is okay. And of course, neurons, they don't have infinite energy. They've got finite resources. So you might want the neural activity to be as small as possible. You don't want big firing rates. You want smaller firing rates. You want to be selective about when you do fire. So again, so this is exactly what we're going to try to think out. I'm going to try to codify these constraints in maths. We're going to almost try to axiomatize a neural representation. I'm going to see what we get. Okay. Okay, so let's think about the first one. Neurons have to be useful. Okay, so we can just come along and say, right, I'm going to assume I have some neural representation that is composed, that is made up of some random variables. So either it's going to be a vector of IID random variables. And then I'm going to believe that my neural representation Z is some linear mixture, some affine transformation of these random variables. Okay, and so this is going to set up, I'm just going to whiz through the math. There's no real math. I'm just saying that it's basically, I'm just saying it's, well, there is math, but I'm just not going to present it here. But I'm just going to say the statement. That means we can just set up a constrained optimization. We want to minimize the energy, the active firing activity of this representation, such that all neurons are positive. And we're going to add another constraint on that the variance in the population is fixed, i.e., so we can't just squish everything down to 0. We can do that, and we can have a look at the form for this this fire overall activity, and we can see that it's equal to a constant, and also, which we don't need to worry about these constants, but it also is also equal to this term. And what this term is essentially telling us how those random variables were mixed in our representation. And so it says, right, we're going to penalize you if you have a neuron. So let's think about neuron J here. Neuron J, if it receives information both from random variable K and random variable L, that's an issue. But if it receives information from K and another K, that's totally fine. So what essentially we're saying is that a neuron in our representation will be penalized if it cares about more than one random variable. That is an issue. This is what just from those axioms, this is what we get. And so the best way we can optimize this neural representation is for each neuron to only care about a single random variable. That is disentangling. So a bit of intuition about why that comes about. So let's say, for example, we have a representation that just cares about 2 of these random variables, and it's going to be random variable E1 and E2, and so that says that we're going to have activity that lies in this square. Okay, so the first thing we want to do is we want to make it positive, so we need to shift that square up to the positive orthant, and then that's a bit of an issue, but not an issue. That's great, but then we want to make it minimize its firing activity. But that's a bit of a pickle. So stuff up here, stuff in the top right is bad for firing activity. A lot of that because it gets squared. And so the best thing you can do is sort of squish that square into the most compact way into your positive orthant. And that, when it's a square, that is when M1 is axis aligned to the x-axis and M2 is axis aligned to the y-axis, i.e. different neurons care about different factors. Disentangling. Okay, so that's a bit of a proof about how a mathematical proof of why you actually get disentangling just from these biological constraints. Quite amazing. Okay, so now we're going to go about and start simulating this and see what happens. But first, we're going to all we're going to do is we're going to change those constraints. I'm going to put them into regularizers. Okay, so we're going to have a non-negativity regularizer, which says all this says is I do not want my representation to, I don't want any neuron in my network to be negative. We're going to have an activity one which says I'm going to penalize you if you've got high activity, and we're this is something I haven't spoken about, but we need to also add a weight activity just so we can't send weights to infinity and activities to 0. Okay, and what we're going to do is we're going to take some training data where we're going to have a bunch of 6 factors. They're denoted by these 6 colors. Okay, when we're going to have input data, which is those 6 factors mixed. Output data is also those 6 factors, but mixed in a different way. And so there is a linear transformation from X to Y. And so we're going to also have a prediction loss on our constraint. This is the functional constraint. It needs to do something. And that's going to say that this network needs to match its prediction of Y to the real Y. And we're just going to train that. And so we're going to train a 1 hidden layer network here. It's a linear network. And so we're going to have, at this point, we're going to turn off all of these constraints. We're just going to have the prediction constraint. And so we've got input goes to some hidden layer, goes to output. And we can see that without these constraints, everything is all mixed and jumbled up in there. And if we look at the matrix of mutual information between neurons in that hidden layer and ground truth factors, we can see that each neuron just cares about lots of different things. Okay, but whereas when we add these biological constraints on there, what we see that individual neurons in this hidden layer, they care about a single factor, and we have a nice disentangling in this middle layer. That was for linear networks. That's where our theory applies to linear things. Oh, sorry, I should say that. Oh, this is just when we're looking at various different combinations of these constraints, and we can see the only two ones that work consistently and well, these red and sorry, these blue and orange ones, and that is the one with all our constraints. Okay, but just done in two different ways. And this, sorry, I should say this is a measure of how disentangled we are. Okay, so now we're going to look at nonlinear networks. This is networks that have they're deep networks, I should say, and they have nonlinear activations as you go through. So and we can say look at how the disentangling works in each of these 5 hidden layers in this network. And so we can see that when we're training on linear data, the same data as before, all of our layers of our network learn nice disentangled representations. But now when we make that data nonlinear, so we're going to keep y the same, but we're just going to apply some nonlinear function to x, or pointwise, or not pointwise, a element-wise nonlinear function to x. And we can see that something really interesting happens. Only the late layers become nice and disentangled, but the earlier ones are really rather entangled. And that makes sense because we need to undo that. We need to undo that nonlinear computation, and undoing nonlinear things requires mixing. But once we've demixed it, then we're back to our nice linear regime, and we can just keep everything nice and unentangled. Okay, and again, we can just have a look at the mutual information matrix, and we see that single neurons care about single factors. Okay, so we can also apply this to unsupervised learning as well. And so just where we're only going to look at observed data, and we're going to try to infer what some latent factors are. And we're going to do this with these autoencoders. Again, so when data was linear, made by linear, a linear process, again, very nice and easy. We extract in the sort of latent layer, we extract factors which are a single neuron only cares about a single factor, and again, that only works for all of our constraints. And again, now in the nonlinear, where we've now created our data from some crazy nonlinear process, from our nice latent factors, from where we know what they are. And then now we come to learn them from an autoencoder. Again, we see that only the case where we have all of our biological constraints, we really do learn these underlying ground truth factors in the world. Single neurons care about single factors. Okay, and we can also do this on standard data sets that machine learners do. So here's a data set where there are lots and lots of different images, and those images have different wall colors, different floor colors, different viewing angles, different shapes, different sizes. And we can try and apply our methods to that. And so what we see is we get very good disentangling, and we get it doesn't look that great here. It's good, but it doesn't look great. But I can assure you it's much better than what other methods get. Even though I've presented this very badly, it makes it look like it hasn't. But I assure you it is quite a lot better. And in fact, all of our results, they sort of fall in this nice Goldilocks area. So what we're seeing here is how well do we fit the data to how well do we disentangle. And there's other methods of trying to disentangle, and they essentially disentangle, but they fit the data badly. But we're all in this sort of nice Goldilocks area of good disentangling, good fitting the data. And so here's an example of what we're seeing. So this is where each latent variable in our model is displayed here. And we can see that this latent variable seems to care about the size of the object. This one cares about the color of the object. This one cares about the floor color. These two don't really care about anything. Neither do these two. This one cares about the wall color only. This one cares about the shape. This one cares about the viewing orientation. We really are learning these disentangled features. We can also disentangle categories by exactly the same theory, and that's just a bit of intuition here. So now if each one of these colors corresponds to a vector which is only active at one, the orange one is only active at one point, and then another time the green one might be active, another time the red one might be active. So there are a category can only be active. It's a categorical distribution, only one thing active at a time. And so classically, they might just be vectors that point in some space, but of course we want to make them non-negative, and then we also want to make them easy to read out. And so if there's a little bit of noise there, of course we want them to be as far away from each other as possible. And again, that's only possible if their axis aligned. If they are in the positive orthon, it's only possible when their axis aligned. And again, a bit messy, but with only the case where we have our constraints there, do we see this nice single variables care about a single category in this case. Okay, I've actually got an awful lot more, but I'm going to unfortunately miss most of it off. Okay, so relating this back to biology, what can this tell us about our cell types? So if you remember, we had these sort of beautiful grid cells, and these beautiful object vector cells, and these beautiful border cells that all seem to be in different neurons. Surely this theory can tell us a little bit about that. Okay, so we're going to train a task now, and this is a task where the animal is going to run around this room, but it's going to like being attracted to these objects. Okay, it's going to like being attracted to these pink objects. And of course, we're going to train another task where the objects are in different locations. So space and objects are factorized, okay, because objects can appear in different locations, and not just 1 and 2 tasks. It's be many, many, many tasks. So it's been trained in all different or configurations of objects. And we're just going to train it in representation, some internal representation. We want it to predict where it is in space, where it is, where are the objects, and what action should it be taken, okay? And objects and actions are going to be pretty entangled with each other because, of course, you head towards objects, but they're going to be very factorized on space because objects in different environments are in different places. And we're just going to train this representation with our constraints. And in the situation where we have no activation, no nonlinear positive rectifying activation, we find this sort of situation where we get these sort of funky looking cells, which are hard to interpret. But in the case where we have a positive activation, we enforce activities to be positive, we get these beautiful grid cells. One, we get them on their own, and they are always the same for every task. They don't change, independent of where the objects are. But we also have these other population of cells which are not active when there are no objects, and they have these sort of vectorial activations towards objects. These are very much like grid cells and object vector cells in the brain, and they really do form in these 2 discrete modules. So you can see that without objects, there's lots of cells that aren't active, but when we do put the objects in, the grid cells don't change, but the object cells suddenly appear. So we have these modules of cells appearing. And again, that's really not true if you just have linear activations. The last thing that I think I'm going to get to talk about is what happens in the case. Remember, I told you before that there's this weird situation where grid cells start warping. Okay, so what happens in the case we just have an entangled task? We don't factorize space and objects. We entangle the 2 together. So we just have objects always in the same location. That's our task, always in the same location. And we're just training the same representation with the same constraints, but now we don't have factorization. And what do we see? Well, we see this rather funny thing that look a bit like grid cells. The astute eye might notice that these cells, the firing activity is always sort of warped towards the cells. They're always sort of locked, they're phase locked to these cells. And if we can compare that to those grid cells where they have no phase relationship, consistent phase relationship with objects, we see this sort of warping behavior. And again, so in the factorized case, we see these 2 modules. In the warp case, we just see this funny warping, entangled thing. And that's going to be very reminiscent of these cells which warp their firing fields towards objects, just like we've done here. And we can quantify that with this is lots and lots of different models run on the same data. And we can see that only in the case where we have we've got the fixed and unfactorized task do we find this this measure of warping. We don't find it in any other case. Okay, so I guess I just have to stop at the interim summary, which is a bit of a shame. But what we've seen is that tasks, when tasks really are factorized, and we're using our biological constraints, it means that these building blocks of our understanding have to exist in separate neural populations. And this is really quite a cool result. There's literally nothing else in neuroscience which can understand the bulk properties of neurons beyond that. People can understand, the task dimensionality understands the dimensionality of our representation. But this is like some weird fine-grained thing which says, no, there is some relationship there, but really different factors must be encoded in different neurons if you want to generalize. Okay, so I'm going to stop there. The next part of my talk was going to be about, this says about how different factors can be must be represented in different neurons, but it doesn't tell us about anything about how individual factors should be represented. And the second part of the talk was going to be about that, but it will maybe next year. Okay, well, thank you very much. Well, maybe I'll skip ahead. And I should thank various people involved in this. Sorry, Rory. Thank you.